Good morning, everyone. I would like to thank the organization committee for accepting our work with the title Intergenic ORFs as Elementary Structural Models of de novo gene birth and Protein Evolution. During the last few years, many studies in different organisms have given evidence of the de novo gene birth, a process by which new genes evolve from DNA sequences that were ancestrally non-genic. However, this process is not well understood, and several models have been proposed to explain this mechanism. Karvounis et al. In 2012 proposed the protogenes as an intermediate state between a non-genic and a novel genic sequence. The key element for the emergence of a protogene is its translation and the production of a small peptide. If this peptide offers an evolutionary advantage to the organism, then it will be subject to selection and will be established as a de novo gene. Then, the novel gene will continue to evolve towards a longer protein by elongating towards its N or C-terminal. So far, different models of de novo gene emergence have been proposed. The pre-adaptation model supports that only highly disordered sequences, pre-adapted not to be harmful through aggregation, will give rise to novel genes. On the contrary, Karvounis et al. 2012 observed that novel ORFs exhibit higher hydrophobicity and lower propensity for structural disorder. Recently, Papadopoulos et al. In 2020 proposed a TM-first model where the membrane environment provides a safe niche for transmembrane adaptive emerging peptides. Even though controversial, all these models support that the initial de novo peptide, once established, will further evolve towards a more canonical and well-folded protein. Consequently, they all give a central role to the full potential of the non-coding ORFs in the emergence of genetic novelty. As a result, several questions about the role of the non-coding genome can emerge. Concerning the de novo genes, what is the sequence and structural properties of the raw material preceding the fixation of the beneficial ORFs? While concerning the established genes, what is its contribution in protein evolution and structural diversity? Answering these questions would permit us to estimate the potential of the non-coding genome to produce novel protein bricks that can either give rise to novel genes or be integrated into pre-existing ones. Our analysis was conducted on the well-annotated genome of Saccharomyces cerevisiae. The first step was to extract and translate to amino acid sequences all the protein-coding genes and all the intergenic ORFs of more than 60 nucleotides. Then, using the hydrophobic clusters analysis, we characterized the diversity of the fold potential encoded in all intergenic ORFs in order to explore the potential of the non-coding genome to produce novel protein bricks. HCA is a method that, given an amino acid sequence, delineates clusters rich in hydrophobic residues presented in green, red, and blue. The clusters are separated by linkers rich in non-hydrophobic residues. Here we represent the corresponding 3D structure of the sequence, and as you can see, the hydrophobic clusters correspond to regular secondary structures, while the linkers correspond mostly to flexible regions. The combination of hydrophobic clusters and linkers on a sequence determines its fold potential, which can be estimated through the calculation of a foldability score. The HCA score ranges between -10 and +10. Low HCA values correspond to sequences enriched in hydrophilic residues, which are expected to be disordered. High HCA values correspond to sequences enriched in hydrophobic residues with high aggregation propensity, while intermediate HCA values correspond to sequences with an equilibrium between hydrophobic and hydrophilic residues, which are expected to fold in solution. We first plotted the sequence length of CDS, presented in orange, and IGORs presented in purple. With no surprise, the IGORs are much shorter than the coding sequences. In addition, the second graph shows that IGORs contain fewer clusters per sequence. Interestingly, the CDS and the IGORs contain hydrophobic clusters of statistically similar sizes. These permit us to propose the hydrophobic cluster as the elementary building block of proteins, which at the same time are found widespread in non-coding sequences. Surprisingly, CDS are enriched in longer linkers. Whether these longer linkers have been selected in order to favor protein modularity and flexibility, or is simply the fact that long sequences are more likely to contain longer linkers, we do not have the answer yet. And even if the hydrophobic clusters between CDS and IGORFs have similar sizes, they do not have the same amino acid composition. On the graph, you can see the propensity of each amino acid for being in HCA clusters of CDS over HCA clusters of IGORFs. CDS clusters are enriched in polar and charged residues, while the clusters of IGORFs are mostly enriched in strong hydrophobic ones. The charged and polar amino acids generate intramolecular interactions, orienting the fold of the proteins and offering fold specificity. We next use the HCA score in order to assess the fold potential of the peptides encoded by IGORFs. As reference, we used 3 sequence datasets consisting of disordered regions, globular proteins, and transmembrane regions extracted from transmembrane proteins. These last ones are expected to form aggregates in solution while being able to fold in lipidic environments. On the plot, you can see the HCA score distribution of the 3 reference datasets. Disordered sequences in green fall on the left part of the plot, adopting low HCA scores. Transmembrane sequences in pink fall on the right part of the plot, adopting high HCA scores, while globular sequences in gray fall on the central part of the plot, adopting intermediate HCA scores. Based on these HCA score distributions, we define 3 categories of sequences: the disordered, the aggregation-prone, and the foldable ones. As anticipated, the vast majority of the CDS sequences fall into the foldable category, reflecting that being foldable is a trait which has been strongly selected by evolution. In contrast, IGORFs contain a wide range of fold potentials, with strikingly almost 2/3 of the IGORFs falling into the intermediate category. Whether peptides encoded by IGORFs in this intermediate category fall into a specific 3D structure, a partially ordered structure, or a rudimentary fold deserve further investigations. This large foldability diversity made us pose the question whether certain IGORFs, based on their fold potentials, are more likely to give birth to de novo genes. In order to address this question and investigate whether IGORFs that gave birth to de novo genes encoded peptides with different foldability potential from the total set of IGORFs, we tracked back the evolutionary events preceding the emergence of 70 yeast de novo genes by reconstructing their ancestral non-genic sequences. Here we present an illustrative example of the emergence of a de novo gene in Saccharomyces cerevisiae. Based on the alignment of the homologous non-genic regions in the neighboring species, we were able to reconstruct the ancestral nucleotide sequence preceding the emergence of this de novo gene. Then, we translated the nucleotide ancestral sequence into the 3 possible frames and detected the ancestral intergenic ORFs that gave birth to the de novo gene. In the same way, we reconstructed all the ancestral sequences for all the branches of the phylogenetic tree and followed the evolution of our de novo gene. The reconstruction revealed 2 ancestral IGORFs separated by a premature codon stop. Interestingly, 2 nucleotide substitutions led to the appearance of a start codon and the mutation of the stop codon into tyrosine, and led to the birth of the de novo gene in cerevisiae. The 70 de novo genes emerged from a total of 167 non-genic ANC-IGORFs. 16 cases emerged from a single ANC-IGORF, which covers almost all the sequence of the de novo gene, and we call them single ANC-IGORF de novo genes, while 54 cases result from the combination of multiple ANC-IGORFs through frame shift events or stop codon mutations, and we call them multiple ANC-IGORF de novo genes. In the plot, you can see the HCA scores of the proteins encoded by the 70 de novo genes at the bottom, and of the peptides encoded by their corresponding ANC-IGORFs at the top. Similarly to the IGORFs, peptides encoded by ANC-IGORFs display a large range of HCA scores. However, the foldability of the ANC-IGORFs does not follow exactly an IGORF-like distribution, as they are enriched in sequences encoding foldable peptides, and depleted in sequences encoding aggregation-prone ones. This suggests that IGORFs encoding foldable peptides are more likely to give rise to novel genes. Then we wanted to study the relationship between the fold potential of peptides encoded by IGORFs and their corresponding de novo proteins. To do so, we connected the foldability of every parent IGORF with the one of its corresponding de novo gene. Multiple IGORF de novo genes are connected with more than one IGORF, while the colors of the lines correspond to the foldability of each IGORF. The overall relationship is characterized by a funnel shape, revealing that most de novo proteins are foldable, even though they emerge from combinations of peptides with various fold potentials. Does this funnel result from the amino acid mutations which transform non-foldable IGORFs into foldable ones, or is it the effect of combining foldable with non-foldable peptides which leads to the final foldable product? To address this question, we hypothesize that since the divergence of their non-genic ancestor, single IGORF de novo genes were only subjected to amino acid mutations, while the multiple IGORF ones have undergone the effect of combination as well. In order to quantify the impact of these different mutational events on the fold potential of the outcoming de novo proteins, we calculated the correlation between the HCA score of each de novo protein on the y-axis and the peptides encoded by its corresponding IGORFs on the x-axis. We can observe that single IGORF de novo proteins display a strong correlation of HCA scores with those of the peptides encoded by their corresponding IGORFs. This suggests that the structural properties of the peptides encoded by the single IGORFs were retained in the resulting de novo proteins. The correlation is weaker for multiple IGORF de novo proteins, which, regardless of the fact that they are predicted as foldable, they emerge by the combination of IGORFs with different foldability potentials. Interestingly, all foldable de novo genes include at least one foldable ancestral peptide, suggesting that in these cases, combining disorder or aggregation-prone peptides with a foldable one has led to the foldable de novo protein as well. Then, we used ribosome profiling data in order to detect translational events in the non-coding genome. We were able to define two types of translated IGORFs: 1,235 IGORFs occasionally translated with a weak translation signal, and 31 selectively translated IGORFs with a strong translation signal. The first are expected to be short-lived, and some of them may give rise to future novel genes, while the second show a more optimized translation activity, which could be related to the emergence of a functional product. In the graph, we are presenting the same properties as before: sequence, clusters, and linker sizes, as well as cluster number, for all the supplementary ORF categories that we defined. Occasionally and selectively translated IGORFs in yellow and red, IGORFs that gave birth to de novo genes in gray, and their de novo genes in blue. In line with Karbunis et al. 2012, for most of the properties, we observe a continuum from IGORFs to CDS, reflecting the successive stages preceding the emergence of a de novo gene until the establishment of a genuine gene. Strikingly, the HCA cluster size remains in general invariant, reinforcing the concept of hydrophobic clusters as elementary building blocks of proteins. Altogether, these results permit us to propose a model which gives a central role to IGORFs in de novo gene emergence and protein evolution. IGORFs encode a wide diversity of peptides, with the majority of them being able to fold in solution. Upon pervasive translation, some peptides that can be deleterious or simply not beneficial will be instantly degraded. Among the rest, some will confer an advantage to the organism and will be further selected, providing a starting point for de novo gene birth. The starting point IGORF, once selected, is subjected to amino acid substitutions, thereby increasing the overall proportion of hydrophilic residues of the encoded peptide. This could lead to the disruption of weak hydrophobic clusters, transforming them into linkers, and the establishment of intramolecular interactions, increasing the specificity of the fold process and the resulting fold. On the other hand, A stop-codon mutation at the starting point IGORF can lead to the fusion with its neighboring yellow IGORF and the elongation of the peptide. We showed that combining IGORFs encoding for different foldability potentials has low impact on the overall foldability of the resulting de novo protein. We can hypothesize that the newly integrated IGORFs will benefit from the structural properties of the pre-existing IGORF network. The fusion of the IGORFs leads to the creation of a longer linker at the IGORF junction. This can also apply for hydrophobic clusters, even though it seems that long clusters are not selected in CDS. Once again, amino acid mutations towards hydrophilic residues may disrupt long clusters generated by fusion into smaller ones or transform them into linkers. After multiple events of amino acid substitution and IGORF combinations, we obtain a protein which displays the canonical properties of CDS, such as long sequence, long linkers, or enrichment in polar and charged residues, which enable the optimization of its flexibility, the increase in specificity of its folding process and its final three-dimension structure, and finally, participate along with domain shuffling or duplication events in the modular architecture of genuine proteins. Thank you for your attention.